And so let's take a look at the fate of the carbon skeletons of amino acids. Okay, and so the alpha amino acids and so can be grouped into classes according to their end products. You have your acetyl coenzyme A, acetoacetyl CoA, the pyruvate, and your several citric acid cycle intermediates, like of course your succinyl CoA. You have your uh, fumarate, and the rest. Okay, and so let's take a look at this amino acid. And so suppose this is actually the interconnection no, between your what you call this your between your amino acids and your citric acid cycle. Okay, and so from your cysteine, and so will be converted into your pyruvate, and then to your acetyl coenzyme A, and then of course the rest of your amino acids. Okay, and so let's take a look at this. As what we said last time, ayan, so you have your 10 amino acids forms your acetyl coenzyme A. Ayan, so acetyl-CoA or your pyruvate. And so with that, and so for your pyruvate, and so you have your alanine, serine, glycine, cysteine, and your threonine. And so for those that forms acetyl-CoA, you have your lysine, tryptophan, tyrosine, phenylalanine, and leucine. For these amino acids, and so they form your alpha-ketoglutarate. You have your glutamate, your glutamine, your arginine, your proline, and your histidine. For your succinyl-CoA naman, ayan, so you have your methionine, your isoleucine, your valine, and your threonine. For your amino acids that form your oxaloacetate, you have your aspartate and asparagine. Okay, and so let's take a look at these amino acids individually. And so you have your amino acids that form your acetyl-CoA and your pyruvate. And so first on the list, you have your alanine. And so your alanine, and so is a directly, I mean, is a reversibly, and so converted, and so to your pyruvate, no? And I mean, yes, reversibly converted to your pyruvate. And so you have probably here your reversible transamination between the alanine and the pyruvate. Okay, and so that is for your alanine. Okay, and so please check and so on this reaction. And so that is reversible. And so from your pyruvate to your alanine and then reversible. And so again, that is reversible. And so accordingly, this one is your alanine pathway. And so your alanine is formed from your pyruvate in the muscle, no? And so that is formed from your pyruvate in the muscle. And so you have there, okay? And so after, and so from the muscle, and so it will be transported into the liver. And so it will be transported into the liver, and your alanine is reconverted to pyruvate by the use of your enzyme, alanine transaminase, no? And so from the muscle, and so your alanine is formed from your pyruvate, and then it will go into the bloodstream and then be jump off to the liver and so be delivered in the liver, transported in the liver, and then it is reconverted into your pyruvate by the action of your alanine transaminase. And so this pyruvate and so is used in the synthesis, of course, of what you call this new glucose. Okay, and so you have here because accordingly the muscle cannot synthesize urea from amino nitrogen, and so the glucose, and so you have actually there that is involved here is your glucose alanine cycle. And so this one is used to transfer amino nitrogen to the liver. Okay, and so accordingly, when exercising, and so muscle produces large amounts of pyruvate. And some of these molecules are converted to your alanine by transamination reactions that involves your glutamate. Okay, and so that is for your alanine. Then you have here your serine. Okay, and so your serine is converted to pyruvate by your pyridoxal phosphate requiring enzyme serine dehydratase. Okay, and so you have here again your serine, and so it's converted directly converted into your pyruvate by your pyridoxal phosphate. And so this one, and so take a look at this one, and so you have your serine is directly converted to your pyruvate. And so same with your cysteine, no? And so that is for that. Okay. And so take a look here. And so your pyruvate to your alanine. And so this is irreversible, no? And so that is for your serine. Okay. Your glycine. And so your glycine and so can be converted and so to serine and so by serine hydroxymethyl transferase. And so again, your glycine can be converted by serine. I mean, your glycine can be converted to serine by serine hydroxymethyl transferase. And then, of course, your serine is converted to pyruvate as what we've seen earlier, no? Ayan, so dun sa pathway. Okay, and then you have your cysteine. And so your cysteine is oxidized to your cysteine sulfonate. Okay. And so your cysteine accordingly is converted to your pyruvate in several pathways. But then for this, and so let's take a look at your oxidation of your cysteine to produce cysteine sulfonate. Okay, and so that this is actually the principal pathway of cysteine. Okay, and then your pyruvate is then produced after the transamination and desulfuration processes. Okay, and so you have your cysteine sulfonate, and so transaminated and remove the sulfur is removed, and to produce your pyruvate here. And then you have your threonine. Your threonine, that is a major degradative pathway. And so in the major degradative pathway accordingly, and so your threonine is oxidized by your uh, dehydrogenase. And so by your threonine is oxidized by your threonine dehydrogenase. And so that is oxidized by your threonine dehydrogenase to produce alpha amino beta ketobutyrate. Okay, and so you have there your alpha amino beta ketobutyrate. And so and this what we call this alpha amino beta ketobutyrate, and so that is metabolized to form a pyruvate. Or and so this alpha amino beta ketobutyrate is cleaved. Ayan, so is cleaved by your alpha amino beta ketobutyrate lyase to form acetyl coenzyme A and glycine. Okay, and so again, ayan, so your threonine is oxidized by threonine dehydrogenase to produce alpha amino beta ketobutyrate. Okay, and so this alpha amino beta ketobutyrate is what they call this further metabolized to form a pyruvate. Ayan, so this alpha amino beta ketobutyrate is cleaved by your enzyme alpha amino beta ketobutyrate lyase to form your your acetyl coenzyme A and glycine. And so that is for your threonine. Okay, and so you have here alternately, and so your threonine can be degraded to alpha ketobutyrate, and so by your threonine dehydratase, no? And so by your again, and so this threonine can be degraded to alpha ketobutyrate by threonine dehydratase, and subsequently to your propionyl coenzyme A. And so subsequently to your propionyl coenzyme A. And so you have your alpha ketobutyrate is what we call this the product, and then this alpha ketobutyrate is what we call this further metabolized to produce your propionyl coenzyme A. And this propionyl coenzyme A is then converted into your succinyl coenzyme A. Okay, and so this is further converted to your succinyl coenzyme A. And then you have your lysine. Your lysine, and so that is converted into your alpha ketoadipate. And so that is your alpha ketoadipate. No, and so that is in a series of reaction. And so actually you have here that involves what they call this two oxidation and removal of your side chain amino group. Ayan, so with that, and then transamination. And so you have your 2-oxidation and removal of your side chain amino group and transamination. Okay, your acetyl acid, your your aceto-acetyl-CoA, and so your aceto-acetyl-CoA, and so your aceto-acetyl-CoA is produced, and so in the reaction that includes several oxidation and decarboxylation and hydration. And so, and this aceto-acetyl-CoA, and so your alpha-ketoadipide, and so is oxidized and decarboxylated and then hydrated to produce your aceto-acetyl-CoA. And this aceto-acetyl-CoA can be converted to acetyl coenzyme A. And so that is your aceto-acetyl-CoA can be converted into acetyl coenzyme A. And so that is for your lysine. Okay, and so you have your tryptophan. Your tryptophan, and so this is your tryptophan is converted to alpha ketoadipate. And so it has actually to convert alpha tryptophan to alpha ketoadipate, you need 8 reactions accordingly. And so you have there, and so it what they call this yield your alpha ketoadipate. And so will further be metabolized to produce your formate and alanine, no? And then your alpha ketoadipate forms your acetyl-CoA, and then this ayan, so your alpha ketoadipate, ayan, so forms your acetyl-CoA, same in your lysine, and then the alanine that is produced in this, ayan, so is converted to acetyl-CoA via pyruvate. And so you have here. Okay, and so your alpha ketoadipate and so forms your acetyl-CoA. That is the same reaction in your lysine and your alanine. The alanine is produced to, is further metabolized to produce acetyl coenzyme A. And then your tyrosine, and so that is for your tyrosine. Ayan, so actually it begins with a transamination and dehydroxylation to produce your homogentisate. And so your transamination and decarboxylation produces homogentisate. Okay, and so this homogentisate is converted to malil malil acetoacetate by your enzyme homogentisate oxidase. Okay, and so this in the absence of your homogentisate oxidase, and so there will be you have here your there is what you call this a disorder related to this one, no? And in this acetoacetate and your fumarate are actually formed from your malil acetoacetate. And so your acetoacetate are actually and your fumarate are formed from what you call this isomerization and hydration reactions. And so you have here, these ones actually are intermediates in your citric acid cycle, your fumarate, no? Then you have your phenylalanine. You have your phenylalanine, and so that is hydroxylated to form tyrosine. Okay, and so your phenylalanine, and so that is hydroxylated to form tyrosine by your phenylalanine for monooxygenase. Okay, and so your the enzyme that catalyzes your hydroxylation of phenylalanine to produce to form tyrosine is your phenylalanine for monooxygenase. Okay, and so it actually requires oxygen and tetrahydrobiopterin, and so your BH4, no? Ayan, so you have there. And then your tyrosine is degraded to form your acetoacetate and fumarate. And so same with your the process that is undergone by your tyrosine. Okay, and then you have your leucine. And so your leucine, and so this is actually one of your branch chain amino acids, and this is converted into your what they call this HMJ coenzyme A. And so your HMJ here is this one is your hydroxy what do you call this? Hydroxy beta methylglutaryl coenzyme A. Hydroxy beta methylglutaryl coenzyme A. Your HMG coenzyme A. Okay, and so your what do you call this? Your leucine is converted to HMG coenzyme A in a series of reaction, and so it includes your transamination, oxidation, and carboxylation, and then finally for hydration. And so this HMG coenzyme A, or your hydroxy beta methylglutaryl coenzyme A, is converted to acetyl coenzyme A and acetoacetate. And so this HMJ coenzyme A is converted to acetyl coenzyme A and acetoacetate by your HMJ coenzyme A lyase. Okay, and so you have therefore your leucine. And so let's take a look at those that form a glutarate. Okay, those that form alpha-ketoglutarate. Okay, and so you have here your glutamate and glutamine. Your glutamine, and so your glutamine is converted to glutamate by glutaminase. And so not only actually glutamate, and so with the action of your glutaminase, and so your glutamine produces glutamate and ammonium ions, no? And so your glutamate is the product, glutamate is converted to alpha ketoglutarate by the action of your glutamate dehydrogenase or by transamination in the muscles. Okay, and so let's take a look at your arginine. Your arginine, ayan, so your arginine is cleaved by arginase, no? And so your arginine is cleaved by your enzyme arginase, and so your arginine is cleaved by your enzyme arginase to produce ornithine and urea. And so recall in the urea cycle, urea synthesis, and so that you have your arginine is involved in there to produce your ornithine and urea. And so you have there. Your ornithine that is produced is converted to glutamate G semialdehyde. Okay, and so this ornithine, and so is your ornithine produced is converted to glutamate G semialdehyde, and then this glutamate G semialdehyde, and so when hydrated and oxidized, ayan, hydrated and oxidized, and so when you subject to hydration and oxidation, your glutamate G semialdehyde will produce glutamate. And so it will produce glutamate. Your what you call this, when your glutamate, and so is when your glutamate undergoes deamination by transamination or by the action of your oxidative deamination, it will produce alpha ketoglutarate. And the other product, urea, and so this urea is, of course, excreted out, and so it goes into the circulation and then filtered out and then eliminated by the urine. Okay, and so that is for your alpha ketoglutarate. And then you have your proline. Your proline, and so your proline, and so your proline metabolism begins with an oxidation reaction. And so your proline, when oxidized, and so forms your D1 pyrroline. Okay, and so this D1 pyrroline is converted to glutamate G semialdehyde, and this glutamate G semialdehyde, when oxidized and hydrated, and so produces glutamate. And then, of course, this glutamate, and so will undergo deamination to produce your uh, alpha ketoglutarate. And then you have your histidine. And so your histidine is converted into glutamate in actually four reactions. No, you have your non-oxidative deamination, and then two steps of hydration, and then removal of the four of the amino group. Okay, and so that is for your histidine naman. Your succinyl coenzyme A. And so your methionine. And so your methionine degradation begins with the formation of your SAM. No, you have it as your S-adenosyl methionine. Ayan, so you have there. And so this one is your formation of your adenosyl methionine is followed by the methylation reaction. Ayan, so that is followed by your demethylation reaction to produce S-adenosyl homocysteine, no? Ayan, so you have there. And then this S-adenosine homocysteine, ayan, so is hydrolyzed to produce adenosine and homocysteine. And see, this one is hydrolyzed to produce adenosine and homocysteine. And this homocysteine, ayan, so this one will is your what you call this homocysteine is metabolized to produce your alpha-ketobutyrate, your cysteine, and actually ammonium ions. And then this alpha-ketobutyrate, and so your alpha-ketobutyrate here is converted into your propionyl coenzyme A. And this propionyl coenzyme A is converted to succinyl coenzyme A. And actually, you have still three steps of what you call this process here to convert your propionyl coenzyme A to succinyl coenzyme A. Okay, and so that is for for your methionine. Okay, and so that is for your methionine. Then you have your isoleucine and valine. And so accordingly, and so these are the processes that will under that the processes for the conversion of this one is actually the same. And so but then and so for your isoleucine kasi and so it is both glucogenic and ketogenic. And so for your isoleucine and so it is your isoleucine. Ayan, so your isoleucine is what you call this converted to produce your acetyl coenzyme A and propionyl CoA. And this propionyl CoA will is converted to produce your succinyl coenzyme A. Okay, and then you have your valine, your propionyl coenzyme A. And so your valine is converted to your propionyl coenzyme A, and your propionyl coenzyme A is converted to your succinyl coenzyme A. And that is your valine is glucogenic only. Okay, and so that is for your amino acids. Okay, and so with that, and so go over your what you call this, your pathways, your Krebs cycle, and then your products of this metabolism of these intermediates of this or the products of these metabolisms of these amino acids, and try to take a look at the what you call this relationship, no? And so please do that. Last, you have here your amino acids that form your oxaloacetate. And so with this, you have your 2-aspartate and asparagine. Okay. And so for your aspartate, and so your aspartate actually is converted to oxaloacetate by single transamination. And so this one, both of them, both of your aspartate and asparagine again are degraded to produce your oxaloacetate. But then, and so your aspartate, and so that is converted to your oxaloacetate by single transamination reaction. No, single. Now, while for your asparagine, and so your asparagine, and so it is first hydrolyzed to produce aspartate. And so you hydrolyze first your asparagine to produce aspartate, and this aspartate is converted into oxaloacetate, no? Ayan, so this is hydrolysis of your asparagine to produce aspartate is catalyzed by your enzyme asparaginase. And so not only aspartate, but then your ammonium ions also. And so the enzyme that is responsible in the hydrolysis of asparagine to produce aspartate and ammonium ions is your asparaginase. And so again, and so please take a look at the connections between these products or the intermediates in this in the series of reactions in your Krebs cycle. Okay, and so that is for your amino acid metabolism.